Thank you very much. Good day, everyone. My name is Ellah Liwonde, and I will be presenting on estimating aflatoxin in farmers' initial groundnuts by testing various groundnuts-grade components in Malawi. I'm currently a master student studying food science and technology at Lilongwe University of Agriculture and Natural Resources. This work was supervised by Dr. Mamadou Ba and Dr. Kasapira, and it was mentored by Professor Whittaker and James Lords. Next slide, please. Yeah, the video we just watched, it's just to give us a clear picture on how it is really a challenge to maintain the quality of groundnuts in shell just after harvesting. As you can see that the nuts were wetted, that water was used to wet the nuts, so that when they're packing into the sacks, you can see that that guy in lead is holding a big stick to squeeze the nuts in the sack, so that they should occupy the more nuts than usual. However, this rewetting, it's a source of further aflatoxin contamination. Next slide, please. Currently, here in Malawi, most groundnuts are traded shelled. So this presents a significant challenge to maintain high quality groundnuts with low aflatoxin concentration. Also, here in Malawi, most groundnuts are shelled by hand. So often they soak the groundnuts in mostly they soak the groundnuts in hot water, so that they should soften the shells to facilitate the shelling process. This rewetting, it's also a source of further contamination, just like the video we have just watched. We all know that the shell protects the quality, the shell protects the crop from insect and pest damage, handling breakages, and further aflatoxin contamination. However, trading groundnuts in shell presents a big challenge for the buyer, since the shell conceals the quality of the groundnuts. To add on that, there is also limited local precedent for this kind of trading, and there is no any systematic grading system to differentiate the good nuts from the bad nuts. So this gives a negative motivation to the farmers for them not to properly manage their groundnuts in the field or even after harvesting, so that they should be able to maintain the highest yield and the highest quality, as well as to reduce the aflatoxin concentration. Another big challenge, which is also a big challenge associated with the aflatoxin testing itself, is the extreme heterogeneity of contamination, whereby the distribution of aflatoxins among the kernels is a skewed one rather than a normal predictable one. So rather than a normal predictable one. Due to this distribution problem, it requires someone to sample a larger sample than the required one, so that the result should hold the truth and to reduce those misclassification. And here in Malawi, we know that over 90% of over 90% groundnuts which are produced here are produced by produced by smallholder farmers who mostly produces smaller volumes. It's not feasible to take a larger sample from such kind of volumes. So there has been an alternative sampling strategy to sample only the components with the high risk of contamination. This is based on the work of Wittig and others in 1998. So in this project, we are opting to adapt the knowing buying point system so that we can segregate the bad nuts and good nuts to determine the quality of the nuts. Next slide, please. The main objectives of the project is one, to verify and evaluate the risk component strategy for small lot sampling. The second objective is to evaluate the knowing buying point system for quality prediction. The third objective is to validate the use of mobile assay aflatoxin testing equipment here in Malawi. And lastly is to provide support training of good sampling and analysis techniques for all technicians using this system. Next slide, please. Yeah, so the hypothesis, we expect that the risk component analysis in groundnuts can be used to determine the aflatoxin concentration for the whole lot for small lot sampling. And the second hypothesis is that prediction of groundnuts grade using visual assessment is ineffective. And lastly, we anticipate that the results obtained from the mobile assay system will not vary from those obtained from the HPLC system. Next slide, please. This is the sampling process. 23 lots of farmers' nut and shell were identified to be sampled, where we collected 95 samples, 95 representative samples aggregated from different parts from the lot. Each sample weighed 3 kgs, and all the samples were transported to Pixar's lab where all tests were conducted. Next slide, please. Let me quickly pass you through the knowing buying point system. The knowing buying point system is composed of a sheller, a shaker table, and a mobile and a quantitative mobile assay system. And it's also composed of a moisture analyzer to test the moisture percentage in the groundnuts. Next slide. The scheme of sample preparation in front of us here is showing us how we prepared all the 95 samples. So three initial groundnuts were first verified to see if it's really weighing 3 kgs, then inspected for loose shelled kernels where they were separated and put in a separate container. Thereafter, all the groundnuts were shelled, then the hulls were discarded. After shelling, all the groundnuts were passed through a shaker table with the two different screens with different sizes. And the third bin, it corrected the third bin, it was the one which corrected the immature kernels and the splits. So in the first screen, we are able to get the sound mature kernels where moisture was tested for each sample. Then on the second screen, we obtained the shriveled kernels. Then each of the three screens were then sorted for insect and pest damaged kernels and model kernels. They were all put in separate containers. So in total, we had 5 different components, which were then weighed and grinded separately. Then aflatoxin was extracted from a 20-gram sample, from 20-gram ground sample. Next slide, please. On the current results, just take note that all the results which I'm presenting here, it's an average of all the 95 samples which I had. The first component, the sound mature kernels and sound splits, we can see that it recorded the lowest aflatoxin concentration. However, it had the highest, it contributed to 71.4% of the total mass of the sample. And it was seconded by shriveled or immature kernels, which had a total aflatoxin concentration of 75.3 parts per billion, followed by the model kernels, which had 295 parts per billion. The highest aflatoxin concentration was obtained in insect and pest damage, where it recorded aflatoxin concentration of up to 418. And lastly, the loose-shelled kernels recorded aflatoxin concentration of 116 parts per billion. The last column here is the aflatoxin mass. This was just calculated by multiplying the mass, the component mass, plus multiplying by the aflatoxin concentration. The total sample here, it was calculated by adding all those components. So it gives us an aflatoxin concentration of 44.3. And just to add on that, we can also see that the shriveled or immature kernels gave us a higher aflatoxin mass of up to 27,842. And the lowest aflatoxin mass was obtained in the loose-shelled kernels, where it gave us a 2,156. Next slide, please. Yeah. After that, I also did a correlation analysis to see if there's any relationship between the grade components and the total sample itself. So as you can see, this correlation is between the loose-shelled kernels and versus the total sample. We can see that we did not have a good correlation. R was equal to 0.85. Next slide. And this is a correlation between the insect pest damage versus the total sample. R, we also didn't have a good correlation. However, R increased. It was 0.392. Next slide, please. This is the chart, a graph showing us the correlation between the model and the total sample. The model kernels aflatoxin concentration with the total sample. We can also see that we did not have a good correlation. However, R increased, and it was 0.417. Next slide, please. That's the correlation between the sound mature kernels and the total sample. As you can see, we also didn't get a pretty good coercion, R was equal to 0.431. Next slide, please. Yeah, that's the graph which is showing the correlation between the immature and shriveled kernels versus the total sample. Just looking at the graph, we can see that we had a very good correlation, and R, it was equal to 0.945. This is just telling us that among all the components, we can use the shriveled and immature kernels to predict the aflatoxin concentration of the total sample of the total sample of the whole lot. Next slide, please. Yeah, so this is just a graph which is showing the correlation between the total sample and adding all the components apart from the sound mature kernels and the sound splits. So we can see that here we also had a pretty good correlation when we add all these components together. So the correlation was 0.969. However, we cannot use this to predict, but use the shriveled or immature kernels, because that component itself, it is already giving us a pretty good correlation to predict the aflatoxin concentration for the whole lot. Next slide, please. The table 2 below is showing us the relationship between the shelling out 10 and the grading at buying. Normally, the grading criteria which was used to grade these nuts, it was a visual assessment. Just looking at the nuts, if you see that there are more visible pops in the groundnuts, it was graded as grade C. And if you can see the pops, but not so much, it was graded as grade B. And if you can see no any visible pops, it was graded as grade A. So, and in terms of pricing, the price was also different depending on the grade. Grade A, it was bought at a much higher price than grade B and grade C. So so the graph you can see, so we also anticipated that for grade A, the shell out 10, it's supposed to be greater than 65%. And for grade B, the shell out 10 is supposed to be between 55 to 65%. And for grade C, the shell out 10, it should range between 45 to 50 to 55%. So when we can see that 21 samples, I had a total of 95 samples. So the shell out range between 41 to 71%. So I had 21 samples which were graded as grade A, but out of the 21 samples, 15 samples, the shell out 10, it was less than 65%. This is telling us that there was a false negative rate of 71%. And only 6 samples out of 21, they were really greater than 65%. For grade B, 1 sample was less than 55%. So this sample, it was supposed to be graded as grade C initially. And for the 3 samples, they were okay. They were supposed to be graded as grade B, but 12 samples were greater than 65%. So this is giving us 13 out of 56 samples were falsely classified as B, giving us a false negative rate of 23%. And on grade C, one sample was less than 45%, and 0 sample was between the range 45 to 55. 17 samples were greater than 55. So this is giving us a false negative rate of 100%. So this is just telling us that for grade A, it was the buyer here who lost. And for grade B, it was okay, but for grade C, the seller is the one who lost here. Next slide, please. Yeah, so we anticipate that if a sample is graded A, grade A, it should be less than 5 parts per billion. And if it is graded B, it should be, after all the sorting, it should be graded between, it should be 6 to 11 parts per billion. And for grade C, after sorting, the end result of the aflatoxin concentration should be between 11 to 20 parts per billion. So here we can see that 6 samples which were graded as grade A, they were really less than 6 parts per billion. However, 15 samples out of the 21 samples were greater than 5 parts per billion. This is giving us a false negative rate of 71.4%. And on grade B, 1 sample was between 6 to 11 parts per billion, which was correctly classified as grade B. And 55 out of 56 samples were the aflatoxin concentration after sorting, it was greater than 11 parts per billion, giving us the false negative rate of 98.2%. And for grade C, 14 samples after the sorting, they were less than 11 parts per billion. This is just telling us that 14 samples were graded as grade C, but they could rather be on grade B or grade A. So this is giving us a false negative rate of 72.2%. And here only 4 samples were truly classified as grade C. So the two tables which we have just seen are just telling us that the visual assessment is not ideal, both looking at the value, the shell out 10, and looking at the aflatoxin concentration after sorting. Next slide, please. Yeah. Unlike the first grading criteria, most samples, I had 56 samples on grade B, and on grade A, I had 21 samples, and on grade C, I had 18 samples. So this is how this graph is showing us how the normal distribution of the grades depending on aflatoxin concentration would have been. So we could have had more grade C samples, then we could have had lesser grade B samples, then fewer samples would have been that of grade A. Next slide, please. In short, we have seen that the aflatoxin concentration of the immature or shriveled kernels is the best predictor to determine the aflatoxin concentration of the whole lot of farmer stock. And we have also seen that the visual rating of the grades was inaccurate. Next slide, please. Next steps, we had to validate the mobile assay with HPLC at Chiteze, and two will provide a grading criteria and purchasing recommendations. And lastly, there will be training for buying center managers and technicians on proper sampling and grading and analytical procedures. Next slide, please. Yeah. COVID-19 has also affected us. We could have already had the validation results, but we are unable to purchase the strips on time. We'll start this early September. Next slide, please. Yeah, thank you for your attention.
Great, thank you so much, Ellah. That was a nice presentation. There are a few comments from Isaac in the chat. A few of them relate to, which I think is a useful reminder to all of us to make sure that, you know, our hypotheses and research questions and objectives are phrased as appropriately as possible. So you may want to just take a look back at your hypotheses and the definition of a hypothesis and make sure that they match. And he also has some, I think, useful advice around your presentation of results in the tables and right around the use of statistics that, of course, we also have to dive into the reasons behind the results that you're seeing. So you may want to take a look at that later. He also asked why you preferred to go for in-shell instead of shelled peanuts in your study. So maybe you can respond to that.
As I showed you earlier that you saw a video, in-shell groundnuts, mostly they handling after harvest. Many people they add water. So this gives the challenge to maintain high quality nuts with low aflatoxin. And again, there is no any grading system which is there to differentiate the good quality nuts from bad quality nuts on the market. So we are to provide that in this project.
Good, thank you very much. Just a note for me that I really enjoyed the inclusion of the video. I thought that was a nice addition and definitely gave a lot of good context for the research that you're doing. So thank you for that.
Thank you.
If there aren't any burning questions for Ellah, then we are running a little bit behind on time, and I know it's been a long session today. So we will move on to her colleague, Michael, who is also working in Malawi on sustainable intensification of groundnuts. So thanks, Ellah, and off to you, Michael.
Thank you.
Good afternoon, guys. This is Michael Meya. As you have already introduced me, I'm doing masters in agronomy. And my academic, I don't know if you are there.
Yeah, you're doing good, Michael. I think somebody else has got their microphone on, so just make sure that everyone's got their mics turned off, and then you can go.
Right. This is Michael Meya from Malawi. being supervised by Dr. Mamadou Ba, and I'm much interested in sustainable intensification of groundnuts production in Malawi. Actually, I'm identifying key entry points. Next. Groundnuts in Malawi is very important, and it's a food legume, even in the world, and it's the third largest oilseed after soya bean and the cotton seed globally. And additionally, it's fourth most important source of edible oil, that is 40 to 50, and third most important source of vegetable protein, and which also contains carbohydrates, especially in sub-Saharan Africa, including Malawi. Next. You know, in Malawi, there is a problem that has to be solved. And you find that yields are very low compared to other countries, especially within the sub-Saharan, even the world. That is due to poor agronomic practices. That is in terms of population and maybe a pattern. And there's also a problem of soil fertility. And even also, there's a problem of cost of inputs, whereby some inputs are very expensive. And on the farmer perspective, you find that they consider best agricultural practice as extra expenses, and they are also labor demanding. And also smallholder farmers, they lack information about the beneficial effects of these technologies, because you find that the scientists are busy maybe in a certain place getting the data. But with this study, you find that it's on farm, whereby the study is within the smallholder farmer perspective. And the researcher challenges to determine how sustainably to improve yields by combining the most effective inputs and farm practices so that farmers, they may have a sustainable return on investment. Next. By looking on the objectives of this study is to evaluate the effects of input combination and farming practices on yield and yield components. Specific objective is to evaluate the effect of input combination and farming practices on yield and yield components. And after the researcher, there is that the combination of inputs and farming practices increase yield and yield components. It's what the researcher is expecting to get from the study. Next. Here is the treatment description of the study. At the first, if you go to the farmer, you find that the first treatment is a farmer practice whereby the plants, the crops are in a single row, and seconded by double row. That is an increasing planting population. And next is a double row applied with inoculant. And there's also double row with inoculant and fertilizer. And we have also double row. We have now 3 inputs. That is inoculant, fertilizer, and gypsum. And also the last is double row and fertilizer. Next. To have a a preview of the plots that we had, you find that the plots were planted with the same rods and the variety, that is CG7. And you find that each treatment has got 4 rows, 10-meter row with 4 treatments and with a control. And the soil samples were collected from each plot. And you find also that inoculant was applied at a rate of, not at a rate of, but 50 grams per 10 kg at soil. So for a farmer to get 1 hectare, he needs to have 8 packs, because 1 hectare requires almost 80 kgs of CG7. So fertilizer application, the farmer needs to use cup number 8, which is 7.5 grams. That is applied at planting, which means per hectare he needs to have 2 bags or 50 bags of 50 kgs, that's per hectare. You may be also interested in knowing the type of fertilizer which was used in the study. So you find that there is 6% of nitrogen, there is 20% of phosphorus, 26 potassium, and 3% sulfur, 0.1 boron, and 0.5 that is zinc. And also you find that there's 13% of lime to deal the issue of soil pH. That's the type. And gypsum was applied at the rate of 15 grams. That is twice the cup of fertilizer. And it was applied at 15 grams per meter by creating a groove between the planting stations. And it was applied at first flower, the first flower. Next. This is the location where the on-farm trials were. It's in the central Malawi and in the northern. And in the center we had, if you can see on the map, there's Kasunga and Lilongwe. And in the north, there's Zimba, Zuzu, and Lumpi. Next. This is the table of results. I was much interested in looking at what is happening in terms of partly cost per hectare. You'll find that if the farmer practice, it will need a seed equivalent to $88.23 US. But when a farmer opts to adapt double row, there's a double in terms of seed, which means if he had 1 pack, he needs to have 2 packs in order to attain that. If he opts to go for double plus inoculant, you'll find that he has double the seed pack plus inoculant. So that's the prices. And if he opts also to go for double row, inoculant and fertilizer, there's also an extra of fertilizer. And when he opts to go for inoculant, fertilizer, and gypsum, there's also a plus of gypsum and fertilizer on the bottom. Next. These are some of the results of which it is of much interest for our smallholder farmers in Malawi. So I'll start with tripod weight per hectare. According to the results, you find that farm practice, whereby the farmer had single row, you find that if you compare with the double row, you find that there's a difference, of course, but not really significant. And if he opts for double, if he opts to add inoculant to the double, you find that there's a difference compared to the farm practice, but it's not really significant, but there's a difference. If he opts to go for inoculant and fertilizer, you find that if you compare with the farmer practice, there's a difference, but not really significant. And if you go for fertilizer, if he goes for double plus inoculant, the all inputs, you find that compared to the farm practice, there's a difference, of course, but not really significant. Next. Here with me again is another yield component, which is of great importance to our smallholder farmers. This is what the researcher found. You find that with the farmer practice compared to double row, there's a difference, of course, but not really significant. If he adds the double to inoculant, you find that compared to the farm practice, there is also a difference, but not really significant. But when he opts for double inoculant and fertilizer, compared to the farm practice, there is a difference. And also when he adds to the previous treatment plus gypsum, there's also a significant difference. This is where now you can solve now the problem. We can go next. Next. As you have seen, it has really showed us that yield and yield components are greatly affected by the combination of the inputs, or even how we practice, how we grow our crops in the field, because these combinations of farm practices will enable the farmers to increase in terms of yield, hence maximize yield as well as returns. But according to the sample size that we had, due to other problems that we have had in the field, with this data that these results that we have at hand, you find that it seems to be good when you go for kernel or dry pod weight. But with the limitations of the sample size, we cannot say or even conclude that they are statistically significant, different, or variations between locations or treatment, because the sample size was so minimal because of other challenges. We have had, amongst the challenges, we had poor seed germination, of which we have been affected heavily. In addition, we have had COVID 19, of which moving around, it was very difficult for us, or even for farmers. And we are looking also to go through the conclusion in order to solve that problem. We need to repetition of the project for the second year, so that if we are going to make conclusions, it has to be very tangible. We are also looking for timely delivery of the inputs to the sampled farmers, because you may find that farmers were failing to grow their crops because some of the inputs were not there at the time. And there's also a need for refresher course for the field technicians as well as farmers, because for the sample to be reduced, there was some problems of which you may find that when you go there, you find another treatment which was not prescribed. For example, the farmer was supposed to have a double row plus inoculant. You find that you have another treatment which is double row plus gypsum. So it has taken us to even reduce the sample size. Next. That is much appreciation to Pixels, Feed the Children and USAID, and even the Peanut Innovation Lab. Next. And next. I thank all for being a good audience during my presentation, and I appreciate for the opportunity being given. Thank you very much.
Thank you so much, Michael. That was a great presentation. I have a couple of questions for you, which I'll ask, and if anyone else has questions for Michael, feel free to either jump in or add them to the chat box. My first question for you was on the costs table. Was that the costs, the sort of economic, the financial costs of buying the inputs, or did you also incorporate the cost of labor?
I did not include the cost of labor. I was looking at the seed only, because there's also another analysis that will be taking place in just near future, of which there's a cost benefit analysis, of which it will include all the inputs that the farmer can use in the field, and then you can compare between the treatments, how much it costs for that particular farmer, and what's the cost that particular farmer can incur on that particular technology that he has adopted.
And my other question for you was, you've mentioned it at the very end, but I know that there were some difficulties in getting farmers to comply with their role in the experiment, right? Can you describe that a little bit more? You talk about needing to do a refresher course. Was it that, did the technicians and farmers not understand what you wanted them to do, or did they choose to do something different?
You know, here in Malawi, you find that the farmers, majority of them, they are much illiterate. You can give them inputs, but they will not be able to follow what has been instructed by the field technicians. You find that during implementing the project in the field, maybe is alone. So instead of putting in inoculant, you might find that he has put gypsum. So that was some of the problems that has taken us to have that minimal sample size.
That makes sense. Thank you. Yeah, I think it's probably something that we've all encountered in our own research. And can you just say, what is your plan to try and avoid that in the next round?
we have to approach the beneficiaries of the project because they are the same beneficiaries that were in this season. So we are going to maintain them, which means the experience that they have, I think they are going to improve from what they had. So we are saying that the refresher course will revamp the thorough understanding of the project. So it will be very easy this time around because they are already experienced with what's the project is all about.
That makes sense. Well, thank you, Michael. I think that we should probably move on to our next presenter, who I believe is Rachel Chemutai, who is presenting her work on enhancing the genetic potential of groundnut in Eastern and Southern Africa. So if you're here, Rachel, off to you. Thanks.
Good day, everyone. Rachel Chemutai is my name. Master's student in plant breeding and seed systems, Makerere University. And the topic of my study is genetics of nutrient-dense groundnut lines for yield and resistance to major foliar diseases in Uganda and Malawi. My supervisors are Professor Patrick Rubahayo, Dr. James Mololo, and Dr. David Kalule-Okelo. Next, please. By way of introduction, groundnuts plays an important role in food and nutritional security among farming communities in the region. It is ranked as the second most important legumes after common beans, and it is a crop that is nutritive in nature. It provides a wide range of nutrients from proteins, vitamins, minerals, and fatty acids. Oleic acid is a monounsaturated fatty acids which is found in groundnuts, and it is beneficial in many ways. It increases the shelf life of groundnuts by reducing the rate of oxidation, thereby reducing their acidity period. And also it has been documented that it has health benefits to human beings by reducing cardiovascular diseases, and also increases insulin sensitivity, and also thereby reduces the occurrence of some cancers. Iron and zinc are the important sources, are the important minerals which is found in groundnut, and also plays a major important role in human health and nutrition by capping malnutrition, especially among young children below the age of 5, and also the women which are under the childbearing age. It also plays an important role in growth and development by playing a role in cellular growth and also cell differentiation. Next, please. Thereby, breeding for enhanced micronutrients, basically iron, zinc, and oleic acid concentration in the groundnut seeds could improve the dietary sufficiency by achieving the required dietary intake of these minerals, and also increases the market value of the product by increasing the premium prices of these products. ICRISAT breeding programs for groundnut have some strides in developing advanced lines which are rich in iron and zinc with oleic acid. But these lines, however, have not been evaluated for performance in the region, especially for yield and yield-related traits, and also to measure biotic stresses. And in this study, we are going to deal with late leaf spot, which is a common occurrence in the region, and also groundnut rosette disease and rust. In the figures, we can see we have figure 1 and figure 2 showing the late leaf spot and GRD. This was an observation that was made in season 2019B in the field. Next, please. The aim of this study is to contribute to development of nutrient-dense lines which are adapted to several agroecologies in East and Southern African region. And this will be achieved specifically by determining the performance of these lines for yield and reaction to major foliar diseases across different environments. This first objective has been partially done. And we have the second objective to determine the effect of genotype and an environment interaction on the micronutrient content in nutrient-dense lines. This study therefore hypothesized that performance of the nutrient-dense lines for yield and reaction to major foliar diseases is similar across different environments. And secondly, there are staple lines for iron, zinc, and oleic acid across different environments. Next, please. The materials and methods that were used were 35 genotypes that were sourced from ICRISAT groundnut breeding program. And we have 2 study sites in Uganda and Malawi. In Uganda, we have Serere and Nakapango, and in Malawi, we have Chitsetse, which are both the hotspots for groundnut rosette disease and late leaf spot. The design that were used were alpha lattice design with 7 blocks and in 5 plots, 2 replication in 2 seasons. The data that was collected was done for disease was done at 4, 8, and 12 weeks, and severity was also scored at harvest. For the scores for LLS and RUST, it is ranging from 1 to 9, whereby it is range whereby less than 3, it shows that the genotype is resistant, whereby 9 is highly susceptible. Next, please. Data was also collected for yield and yield-related traits. We had the 50 days to flowering, and also we had the shelling percentage, biomass, and the number of pods per plant, 100 seed weight, pod yield, and the whole meal. The data that was collected was analyzed in GenStat 18th edition, whereby ANOVA tables and predicted means were generated. Next, please. These are the preliminary results for objective 1, whereby we have the means of squares, and we have a range of a number of variables, whereby it was obtained from the results that the environment did not have an effect on the performance of the genotype for a number of traits, except for whole meal and plot seed yield. Next, please. Further, we have the results for RUST that was done across locations. The blue showing Nakapango, and the orange showing Serere. It was depicted from the results that most of the performing lines was seen from Nakapango with a number scoring of less than 2. And but the lines that were most affected in this RUST were from Serere. It can be concluded in this graphical representation that Serere had a higher rust pressure than Nakapango. Next, please. We also have the results for late leaf spot across locations, whereby most of the lines were moderately resistant to susceptible in Nakapango and in Serere. The ICGV 160 showed a consistent predicted means across locations of less than 5, while ICGX-SM 14033 B61131 had the highest severity across both locations. Next, please. We also have the results for groundnut rosette disease percentage for disease incidence that was calculated. Most of the lines had around 90% of the line showed scored less than 30%. This means that most of the lines are a bit resistant to rosette disease. And we had the ICGX-SM 14026 B101212, which showed a lowest cost for GRD. Next, please. We also did the yield traits, whereby here we have the 100 seed weight across location, which was compared to checks. Here we have the JL24 and ICG VSM 9704 as the checks. And the highest genotype which showed a higher seed weight was the ICGV 171024. This could be used as a confectionery, for confectionery purposes, because at least something which scored above 55 grams can be used for confectionery breeding. Next, please. We also did a number of pots per plant, which was done in Sereda. That was to check for the best performing compared to checks. And we have ICGV 16013 and ICGV 140-3P2 1212 performing a little bit higher than the best yield check, which is ICGV SM 9704. This then could be recommended for best yielding genotypes. Next, please. For a conclusion and way forward for objective 1, the preliminary data, which we already have, identify some resistant lines, which also in which also we got the high yielding ones, which are compared to checks. They can now be used further in the breeding program, and also the nutritional data that will be obtained when we send the samples for iron, zinc, and oleic acid will guide the final selection of these lines that can be further utilized by the program. We will further retrieve the data from Malawi to compare with the data that we have here in Uganda. And we will do also the soil and weather data for the region, East and Southern African region, which we will use to compare for genotype and environment interaction for the nutrient density. We'll further include the data for 2020A, in which the harvesting is already ongoing. Next, please. For objective 2, however, we have had some issues. We have not been able to do the analysis, but the samples have been sent for analysis for this micronutrient content, which is ongoing. And also we haven't done the analysis of soil, which we hope to do it very soon. Next, please. We have, however, had some challenges, like most of my colleagues, due to the COVID. Most of foreign students, including me from neighboring countries, we had to return home. But the environment, however, has not been very much conducive for work and also for meeting, especially when we talk of internet connectivity and even the environment generally. Also, we have it has not been possible to have a direct meetings with our supervisors and also fellow colleagues, which would have helped for further discussion in an analysis of data and other things that we were supposed to do. The laboratory closure also has affected the analysis of the nutrients and also for the soil. Next, please. I wish to acknowledge the Peanut Innovation Lab for this noble opportunity for giving us to participate. Also my APOL supervisors for all the guidance and my colleagues for all the help. Nasari, Ekrisa, True Forum, Makerere, and the SCIPSA for all the support that they have accorded to me. Next, please. And thank you very much for your kind attention. We love peanuts. Thank you.
We do love peanuts. Thank you, Rachel. Does anybody have a question? Yeah, one just came in from Dave in the chat box. So maybe you can take a look at that, Rachel. He's wondering how significant were the yield differences that you observed? And was yield correlated with any of the disease resistances?
Okay, significant what the yield differences was yield. Okay. Basically, the significance were not too much for yield traits. Only I specified that the yield difference that we saw in whole meal and the 100 seed weight. Yeah. And was there any yield correlated with any disease resistance? Yes, I was able to observe that some of the disease or some of the genotypes that were really having high scores of the disease were showing that the yield were a bit lower than the other one, which was having low or moderately resistance. Yes.
Great, thank you so much, Rachel. I don't think that there are any other questions now, and we still have two more presenters that I'd like to get to, but thank you for your presentation. I think for everyone's benefit, we do understand, of course, that COVID has made our work more difficult, and that part of that relates to communication with students. Got their mic on. For students who don't have access to universities and to those networks now, we understand that that's caused challenges, and so just to let you know that we hear you, and that's something that we're working on as a lab to make sure that you have the connectivity you need to complete your studies and your research. So I think that we have two final presenters this morning, Hamady Ba and Mamadou Ba, both of whom are working on the optimized shrub system project in Senegal. I believe that they would like to present at least partly in French today. So so we'll go back to our sort of hybrid language effort here. If you have questions and you do not speak French, then feel free to type them in the chat box in English, and I and the other French speakers on the call will do our best to translate them for our presenters. Ok, bon, je vais essayer de parler en français maintenant pour les francophones ici, et je suis désolée en avance parce que ça fait beaucoup de temps sans l'occasion de pratiquer français. Donc, nos deux derniers présentateurs sont Hamady Ba et Mamadou Ba, qui nous rejoignent du Sénégal et qui font leur recherche sur un système d'arbustes qui peuvent être utiles à faire beaucoup de choses, mais par exemple pour adresser les problèmes de sécheresse dans l'Afrique de l'Ouest. Donc, c'est Hamady et Mamadou Ba qui va commencer. Donc, à vous.
Bonjour. Bonjour. Mon nom est Hamady Ba. Je suis élève ingénieur à l'ENSA et je suis là aujourd'hui pour faire la présentation de mon protocole de mémoire. Il y a pour analyse des pratiques de gestion de la fertilité des sols dans les systèmes de culture d'arachides et de céréales dans les zones de Nioro et Meya. Next, please. Voilà mon plan de présentation. On a la problématique, l'objectif de l'étude, les résultats attendus et la méthodologie. Next, please, next. Oasis. Sénégal, la dégradation des sols pousse près de 2/3 des terres cultivables. Environ 34% de la superficie du pays. À cette dégradation s'ajoutent les effets néfastes du changement climatique. Dans le bassin arachidier, la coupe répétitive des arbustes associés à des pratiques culturelles qui favorisent leur mise en feu ou leur transportation hors des parcelles de culture contribue à la réduction de la teneur en matière organique des sols. Ces pratiques ne favorisent pas la dégradation microbienne de la biomasse dans les parcelles de culture. Les travaux de recherche ont permis de mettre à disposition une innovation dénommée Optimized Service System, OSS, qui favorise la fertilité et la gestion de l'eau afin d'augmenter les rendements des cultures et les revenus des exploitations familiales. En vue d'une diffusion de cette technologie, il convient de déterminer quelles sont les pratiques de gestion de la fertilité des sols dans les systèmes de culture d'arachides et de céréales. Next, please. Next slide, please. Maintenant, on est à l'objectif de l'étude. L'objectif global est d'analyser les pratiques de gestion de la fertilité des sols dans le centre-nord et dans le centre-sud du bassin arachidier. En effet, on a trois objectifs spécifiques. Analyse des systèmes de culture d'arachides dans le centre-nord et dans le centre-sud du bassin arachidier. Ensuite, étudier les pratiques de gestion de la fertilité et enfin déterminer la place des arbustes et de la gestion de la fertilité dans les systèmes de culture d'arachides. Next slide, please. Nous nous changeons au résultat attendu. On a d'abord les systèmes de culture à base d'arachides dans le bassin arachidier sont analysés. Ensuite, les pratiques de gestion de la fertilité dans la zone d'études sont étudiées et enfin la place des arbustes dans les systèmes de culture à base d'arachides et la gestion de la fertilité des sols est déterminée. Next slide, please. On est maintenant à la méthodologie. On a déjà réalisé la revue documentaire au niveau de la bibliothèque de l'ENSA à travers les mémoires, les articles, les personnes et structures ressources, mais aussi à travers les sites Internet avec les moteurs de recherche. Maintenant, il nous reste à faire les enquêtes qui se feront auprès de 300 ménages, c'est-à-dire 150 à Nioro et 150 à Meya. On va les faire à l'aide de tablettes déjà programmées pour recueillir les informations. Next slide, please. En résumé, on va faire les enquêtes de terrain. Ensuite, analyse des données et la rédaction du mémoire et enfin on va faire la présentation du mémoire. Next slide, please. Thank you. Thank you very much. Merci de votre attention.
Ok, très bien. Merci beaucoup, Amadou. C'est évidemment un domaine important de la recherche. J'ai une question ou deux questions pour vous. Première chose, pourquoi avez-vous choisi ces deux différentes zones, les deux différents endroits où vous allez marcher? Et est-ce qu'il y a de grandes différences par rapport au système de cultivation ou d'autres choses?
Oui. Bon, on a choisi ces deux zones parce qu'ils sont dans le bassin arachidier et dans la zone de Nioro, on retrouve les arbustes liositiquement et dans la zone de Meya, on retrouve les arbustes Ngera Senegalensis. Oui. Okay, merci.
Merci. Et dernière question. Pour les enquêtes que vous allez mener, est-ce que vous allez faire les enquêtes en personne ou à cause de Covid, est-ce que ça va créer des problèmes? Est-ce que vous allez faire les enquêtes par téléphone ou qu'est-ce que vous allez prévoir?
Non, non. On va on va les faire en personne même. On va seulement mettre des masques seulement et avoir des gels pour se laver les mains.
Ok. Bon, merci beaucoup. S'il n'y a pas d'autres questions, je pense que nous pouvons continuer avec notre dernier présentateur. C'est Mamadou Ba, c'est un collègue de Amadou, je pense, qui fait la recherche sur le système des arbustes. Donc, merci Amadou, et parce que moi, ma domaine de recherche, c'est le genre. Donc, j'attends avec impatience vos résultats. Merci.
Okay.
Okay, so thanks to Hamady, and our next presenter is Mamadou Ba, who is also working on the same project in Senegal on the shrub system.
Il n'est pas encore en ligne.
Ah, il n'est pas encore en ligne. Ok.
Je suis là.
Ok.
Ok.
Ok, à vous.
Ok, merci beaucoup. Bonsoir à tout le monde. Ou bien bonjour à tout le monde. Je m'appelle Mamadou Ba. C'est avec beaucoup d'honneur et de plaisir que je vous présente les résultats de mon mémoire de fin d'études portant sur l'analyse des déterminants d'adoption de la technologie Optimized Shrub System, cas de Ngera sénégalensis et de Philostigma reticulatum dans l'ambassade arachidier. Sous la supervision de mes encadrés, Dr. Idrissa Wade et Karim Touré. Next, please. Pour mener à bien cette présentation, le plan suivant a été adopté. Donc, une introduction, la méthodologie, les résultats préliminaires, la conclusion et les recommandations. Next, please. Au Sénégal, la coupe répétitive des arbustes, à savoir Ngera sénégalensis et Philostigma reticulatum, contribue à la réduction de la matière organique dans les parcelles de culture. Next, please. Cet état des faits entraîne une réduction drastique des rendements de culture, à savoir le mil et l'arachide dans les ménages, et ce qui expose la plupart de ces ménages à une insécurité alimentaire et à la malnutrition. Next, please. L'innovation Optimized Subsystem prône l'utilisation des Grad sénégalensis et des Pliostigma reticulatum comme agent fertilisant du sol à travers la matière organique. Au lieu d'être brûlé et transporté hors du champ, l'innovation propose une utilisation de cette matière organique, à savoir couper et enfouir cette matière organique pour relever le niveau de fertilité des sols et augmenter à partir de le rendement des cultures et le revenu des producteurs. Next, please. Alors, les bénéfices, l'effet bénéfique des Grad sénégalensis et des Pliostigma reticulatum ont fait l'objet de plusieurs travaux scientifiques, et cela depuis environ 20 ans de recherche. C'est dans ce cadre que l'ENSA de Thiès, en collaboration avec l'Université d'Ohio, ont initié cette étude dont la question principale est de savoir quels sont les déterminants, les facteurs favorisant l'adoption de la technologie dans les deux zones d'études. Next, please. Alors, l'objectif global est de déterminer les facteurs favorisant ou non l'adoption de la technologie dans les deux zones d'études. Donc, il s'agira spécifiquement de faire une caractérisation socio-économique des producteurs, les systèmes de culture, de prendre ou bien d'analyser la perception des producteurs sur les arbustes et de faire une évaluation économique des coûts supplémentaires apportés par la technologie. Next, please. L'étude a été menée dans le bassin nord du arachidier, dans la zone de Meya, dans 6 villages avec 15 producteurs, dans le bassin sud, dans la zone de Nyoro, donc 3 villages avec 15 producteurs. Donc, en tout, nous avons 30 producteurs qui ont des parcelles paysannes et une parcelle où l'innovation Optimized Subsystem est appliquée. Next, please. Alors, au mois d'octobre, nous avons mené notre première enquête. Donc, au niveau des deux zones, les 30 producteurs. Donc, l'objectif principal, c'est de déterminer ou de savoir les rendements de culture de la saison 2018, de voir l'appréciation des producteurs par rapport à la qualité, à la fertilité du sol de leur champ, de voir aussi leur niveau d'équipement, leur méthodologie de fertilisation ou bien de conservation de la fertilité, de déterminer leurs perceptions par rapport à Grad sénégalensis et Pliostigma reticulatum dans leurs parcelles de culture. Next, please. Donc, au mois de janvier, après la récolte, nous sommes retournés au niveau des deux zones pour faire aussi des enquêtes, donc pour déterminer l'appréciation des producteurs de la technologie durant la première année, de voir les contraintes qu'ils ont rencontrées pour la mise en œuvre de différentes parties de la mise en œuvre de la technologie, à savoir la multiplication des arbustes, le coupe ou bien le repiquage de ces arbustes. Et de déterminer maintenant, est-ce qu'ils ont remarqué une différence significative entre la parcelle paysanne et la parcelle de l'innovation. Next, please. Alors, pour les résultats, nous avons constaté que l'âge moyen des producteurs dans les deux zones est de 51 ans. Et l'autre constat frappant, c'est que 63% des producteurs ont plus de 45 ans, ce qui montre la tendance à la vieillesse des producteurs et met en exergue le phénomène d'exode rural des jeunes qui quittent de plus en plus le monde rural pour se retrouver en ville. Next, please. Alors, le niveau d'instruction des producteurs n'influence pas forcément sur l'adoption de la technologie à partir du moment où la technologie peut facilement s'expliquer en langue locale, en Wolof, en Peul, voilà, et en Serer. Donc, ce n'est pas un frein par rapport à l'adoption de la technologie. Next, please. Alors, quant à la perception des producteurs sur l'effet bénéfique ou non de Ngera sénégalais et des pliostigma reticulatorum, ils sont tous unanimes sur le fait qu'ils ont constaté que tout autour de la canopée des arbustes, l'humidité est beaucoup plus élevée à ce niveau qu'à une autre place des champs, et que aussi les cultures se développent plus sous la canopée de Ngera sénégalensis et de pliostigma reticulatum. Ces constats ont été aussi prouvés par les études de Kizito 2007-2009, Kizito et al., de Dick et al. 2009 et de Jeju et al. 2013. Next, please. Maintenant, par rapport à la multiplication des arbustes, les producteurs sont tous unanimes qu'ils n'ont jamais effectué cette activité, qui ne savent pas comment multiplier les longueurs ou bien le pliostigma. Donc, ça, c'est unanimement, aussi bien à Nyoro qu'à Mehe, tous les producteurs sont unanimes qu'ils ne peuvent pas, qu'ils ne savaient, qu'ils n'ont jamais utilisé une multiplication de pliostigma de Ngera. Next, please. Alors, pour calculer le coût additionnel apporté par les activités supplémentaires à l'hectare de la technologie, nous avons prôné deux scénarios. Le premier, c'est de valoriser de façon monétaire toute activité supplémentaire apportée par la technologie. Notre deuxième scénario, c'est de ne pas valoriser cette activité, c'est de faire comme si le producteur le ferait de façon gratuite. Next, please. Alors, il est ressorti que lorsqu'on valorise les activités de multiplication des plants, donc le plan revient dans le scénario 1 à 56 francs CFA et à 10 francs CFA dans le scénario 2. Et donc, je rappelle, au niveau de la multiplication des arbustes, il y a le coût de l'eau, le coût des sachets, le coût de la main-d'œuvre, à savoir le marcottage et le coût d'arrosage. Next, please. Quant à l'activité de répiquage, aussi, donc, il revient à 12 francs lorsqu'on valorise l'activité de la main-d'œuvre, mais lorsqu'on ne valorise pas la main-d'œuvre, c'est 0 francs. Next, please. Maintenant, pour l'activité de coupe, hachage et enfouissement, lorsqu'on valorise l'activité, donc ça revient 12 francs CFA par gain de pliostigma ou de Ngera. Alors lorsqu'on ne le valorise pas, c'est 0 francs. Next, please. Alors, à la première année, il n'y a pas une différence significative en termes de rendement dans les parcelles de culture, aussi bien qu'à Nyoro qu'à Mehe. Donc, il n'y a pas eu de gain net dans la parcelle de l'innovation. Next, please. Alors, pour la deuxième année, dans les scénarios, donc nous avons trouvé dans notre scénario 2, donc à partir de la deuxième année, déjà on commence à avoir une différence de rendement et une différence en termes de gain, donc notamment 20 000 francs CFA à l'hectare. Next, please. Si toutes les activités de multiplication et autres sont maîtrisées, donc il va de choix que la troisième année, donc il y aura une différence significative en termes de rendement. Et ces données sont basées sur l'article d'Ossa et al. Qui ont publié, qui ont fait une étude de 3 ans ou 4 ans sur ces deux parcelles. Alors ça, tout cela, c'est le compte non tenu de l'apport positif, des externalités positives qu'apporte l'innovation à l'environnement, au changement climatique et en tout cas au niveau social, ça on n'a pas calculé ces aspects. Next, please. Alors nous en sommes à la conclusion. Donc la dégradation des sols dans le bassin arachidier, combinée avec la diminution de la pluviométrie et à la mauvaise qualité des semences, ce qui rend vraiment les rendements de saison beaucoup plus petits et les rendements ne sont pas vraiment élevés. Next, please. Alors, durant notre travail, nous avons répertorié ces facteurs comme étant des facteurs impactant l'adoption, à savoir l'âge du producteur, parce que c'est des activités qui demandent un effort physique. Et dans les deux zones, on voit que la tendance à la vieillesse est une réalité. Donc, ça risque d'être un frein à l'adoption de la technologie. L'autre facteur aussi qui risque de ne pas favoriser l'adoption de la technologie, c'est la maîtrise de la multiplication des plants de Ngera et de cliostigma. C'est des activités qui n'ont jamais été faites par les producteurs et qui demandent beaucoup de technicité qu'ils ne maîtrisent pas. L'autre aussi facteur, c'est le suivi de ces arbustes après la plantation dans les champs, parce que durant la saison sèche, il y a la transhumance, il y a le bétail qu'on laisse dans les champs, et lorsqu'il y a une pénurie de pâturage, donc les jeunes plants sont beaucoup plus appétants, les animaux risquent de manger les plants qui sont récupérés, piqués, et engendrant d'année en année un coût supplémentaire. Et aussi, l'autre facteur qui doit être déterminant pour l'adoption de la technologie, c'est qu'il y ait un avantage comparatif entre la parcelle de l'innovation et la parcelle paysanne. Next, please. À l'issue de cette étude, nous recommandons aux partenaires, en tout cas le projet, de consolider les partenariats avec le groupement des producteurs, Symbiose et UGPM, et aussi d'élargir ce partenariat avec les organismes de développement rural, à savoir la DRD, l'ANCAR et les eaux et forêts, de finaliser la fabrication du prototype de machines pour diminuer la pénibilité du travail de coupe, de hachage et d'enfouissement, de créer des vidéos tutoriels pour pouvoir expliquer la technologie et l'élargir à tous les producteurs, et d'utiliser les radios rurales pour la communication et la dissémination à grande échelle. Next, please. Voilà donc qui m'a fait à notre présentation. Merci de votre aimable attention. Thank you for your kind attention.
Oh, super, merci beaucoup Mamadou. Il y a une question pour vous dans la boîte à droite de Page, qui est une demande liée à la présentation de votre collègue. Avez-vous parlé avec des femmes aussi bien que les hommes? Et est-ce que le genre est un facteur dans l'adoption de ce système, selon vous?
Oui, en fait, pour le choix des parcelles, donc la plupart des gens s'adressent d'abord au chef de ménage, parce que c'est eux qui distribuent les parcelles. Donc, dans notre échantillon, il n'y avait que 5 femmes sur les 30. Donc, ce qui montre que aussi, même dans les enquêtes, la distribution des parcelles de culture se fait par les hommes. Et lorsqu'on s'adresse à un ménage, on s'adresse en premier lieu à l'homme, c'est celui qui distribue les parcelles. Donc, c'est pour cela que vous n'avez pas autant de femmes dans notre échantillon. Mais ce n'est pas un facteur, en tout cas, limitant, parce que les femmes, aussi, lorsqu'on les explique la technologie, elles le comprennent parfaitement en langue locale, tout le monde comprend. Voilà. J'espère que j'ai répondu à la question.
Oui, merci. Et pour moi, j'avais une autre question par rapport aux coûts de travail que vous avez calculés. Je voulais savoir comment vous avez calculé ces coûts. Est-ce qu'il y a un standard au Sénégal?
Oui, en fait, j'espère à voir les techniciens qui faisaient la multiplication de guerre ou bien de pilostigma. Donc, il avait répertorié tous les charges qu'il a mis pour produire un plan. Par exemple, l'achat des sachets pour les gaines de pilostigma, la quantité d'eau qu'il a utilisée durant les 6 mois de préparation des pépinières. Donc, cette quantité d'eau, si je le rapporte au mètre cube qui est vendu dans les milieux paysans, qui est de 250 francs le mètre cube, donc ça me donne le coût de l'eau utilisée pour avoir 1200 plants ou 1500 plants. Donc, et aussi le nombre de personnes qu'il a utilisé, la matière organique qu'il a utilisée, tout ça, il l'avait répertorié. Nous avons fait des calculs en se basant sur les prix qui se trouvent dans le monde rural et non les prix qui se trouvent en milieu rural.
Ok, merci beaucoup. S'il n'y a pas d'autres questions, je pense que nous pouvons terminer notre réunion aujourd'hui. I think that we've probably reached an end. We're almost at noon here, at least on the East Coast of the US. So I think with that, we should probably wrap up for the rest of the day. Thank you all to everyone for your participation today and in the preceding graduate student presentations that we've had. I think it's been hopefully, it's been very informative for us at the lab. I hope that it's also been useful for our students to get some feedback, to feel a little bit more included in the work that we do at the lab, and to be connected to students and scientists working across Africa and the United States. So thank you guys very much again. I think that this is something that we hope to continue investing in, these relationships between members of our lab. So stay tuned certainly for other events that we plan to organize with you.